Ayan. Wala na ba papasok? Okay. I guess. Co-host. Meron na kayong knowledge ng biochemistry, tama? Tapos, sana meron kayong good foundation nun kasi ito ay patungkol sa medyo genetics to. So, yung sana maayos yung background nyo ng DNA analysis, RNA analysis, transcription, translation. Sana maayos lahat yon. Kasi kung hindi, mahihirapan kayo dito. Okay. So, first topic for molecular diagnostics ay restriction fragment length polymorphism. So, this section discusses clinical laboratory techniques, katulad nga na sinabi ko kanina, for used in molecular diagnostics for DNA and RNA, and clinical applications for these techniques. Sa dulo natin i-discuss yun. So, first, we will go on the laboratory methods na katulad sinabi ko na. Yung first ay restriction fragment length polymorphism or RFLP. So, for this method, sample DNA is first cut into pieces, into small fragments by endonucleases. So, cut into fragments by endonucleases. Mais ba yung laser? Sana kita. Sana kita yung laser. Ano? Kunwari dito, meron tayong blood stain, pwedeng blood serum, pwedeng plasma. Yung sample DNA, kakat ng to fragments by endonucleases. So, meron tayong iba't ibang type ng endonucleases for various lengths ng fragments para makapag-produce ng various length ng fragments. So, it is important to note that each set of fragment is unique sa individual. So, kunwari, si Mr. Panganiban, kumuha ako ng blood serum niya, nag-cut ako through various endonucleases. Tapos si Miss, sinto? Nalimutan ko yung pangalan. Kunwari si Miss Valia, nag-cut ako ng fragments niya. Unique yun. Kasi, di ba, sobrang haba kasi ng DNA. So, sobrang baba ng possibility na ang two individuals na hindi naman twins or what, ganun, ay sobrang baba ng chance na magkapareho sila ng sequences. Sobrang baba talaga. So, first endonucleases, then yung fragments ng DNA ay separated through electrophoresis. So, ano yung sa electrophoresis? Ay separation through molecular weight. So, yung DNA fragments transferred to a membrane gagamit ng Southern blot. So, application to ng Southern blot na i-discuss natin mamaya. Dapat pala yun yung inuna ko. Then, yung radioactive DNA probe, pwedeng fluorescence or what. Pero dito ay pwedeng gumamit ng radioactive DNA probe. Yun yung gagamitin para makita natin yung mga spots o yung mga fragments. Ma-visualize natin kung nasaan yung mga fragments. Tapos, ang tawag nito, anong tamang term? Ko-compare natin sila with each other para makita natin kung magkapareho. So, kung hindi magkapareho yung mga sequences sa fragments, edi hindi siya yon. Ganun. Kasi ito ay ginagamit to make, nga din dito sa slide, definitive identification of an individual. So, ayun. Ginagamit din actually ito sa forensic chem. Ayan. So, next ay PCR. So, polymerase chain reaction. So, this method uses thermal cycling to amplify or produce many copies of DNA. So, yung things na need natin ay DNA to be amplified, 2 primers, 3 prime end and the 5 prime end, polymerases, nucleotides, buffer, magnesium, and potassium ions. So, para saan yung magnesium tsaka potassium ions? So, pwede yung mag-chelate dun sa phosphate grooves dun ng DNA backbone. Kung naalala nyo, ano nga yung component ng DNA? Ano yung buo niyang pangalan? 2-deoxyribonucleic acid. Tama? Mayroong phosphate group, may ribulose group, pero 2-deoxy. So, walang oxygen dun sa 2 position. Tapos, ayun nga, ribose, phosphate, tapos yung nucleotide base. Okay? Then, dun sa phosphate backbone ng DNA, ginagamit yung potassium or pwedeng magnesium to chelate that para mag-stabilize yung duplex formation. So yung ammonium ion naman can interact with the nitrogenous bases dun sa nucleotide base to destabilize naman. So, ayan. So, step 1 nandito sa slide. Step 1, denaturation step, usually at 95 degrees Celsius for 20 to 30 seconds. So this separates the double-stranded DNA by breaking the hydrogen bonds between the nucleotides. Sir, bakit nagbe-break? Kasi through heat. Naalala nyo anong nag-stabilize sa isang double-stranded DNA ay hydrogen bonding. Tama? Tapos, pwede nating ma-denature through heat. Ang usually 95 degrees Celsius or pwede na yung 94. Ayun. So, second step, annealing step. So, i-lower natin yung reaction temperature to 50 degrees Celsius or 65 Celsius. Depende, or 72, kahit saan doon. 50 to 65 or 55 for 20 to 40 seconds. So, this allows the yung primers to anneal to the single-stranded DNA template. Paano naging single-stranded? Kasi break natin to denaturation, naging single-stranded, tapos i-anneal natin ngayon. So, once the primers anneal, the polymerase begins DNA formation. So, third step, elongation and extension step, or yung step 3, usually at 72 degrees Celsius. So, the optimum temperature ng polymerase na active ay sa 72 degrees Celsius. So, the time required for this step depends on the polymerase and the length of the DNA template. So kung mas mahaba, mas matagal. Kung mas maiksi, mas mabilis. So a good estimate is that the polymerase will polymerize 1,000 to 1,500 bases per minute depende sa machine. Ayan. Pero yun yung standard. So, final step. Hindi pa to final step. Second to the last. Final elongation. Ito makikita nyo sa dulo. Ito, final elongation. Yung repeat cycle ng 20 to 40 times. So to ensure that all single-stranded iDNA has been amplified, so the solution remains at 70 Celsius, 5 to 15 minutes after the last PCR cycle. Basically, pinaparami lang natin sila. Tapos to preserve the DNA, yung tinatawag nating final hold, the DNA produced to PCR, yung solution, i-hold natin from 4 Celsius to 15 Celsius. Indefinite period of time, pwedeng ilagay sa freezer or what for storage, or pwede nyo nang gamitin for analysis. So, that is the polymerase chain reaction. So, may video dito. Punta tayo doon sa video. Panoorin natin. Okay. Stop share. Stop share muna siguro ako. Stop share. Stop presenting. Punta muna tayo dun sa video. Yes, sir. What the heck ads? Okay, andito na. Share ko na lang. Itong YouTube vid na to. Hello? Okay, rinig ba? Lakas ng mga speakers ko. So, ito ay PCR. Pakita niya sa iyo kung paano setup. Ano siya ginagawa. Kasi wala naman tayong PCR sa LSP.
Shopee po kayo, sir.
Ano, paki rinig ba?
Kayo po ang sir, wala pong sound sa akin.
Sa ilan ba walang sound? Ito narinig niyo ako?
Opo.
Okay, email ko lang yung PDF nito, panood na lang nung link. Kasi papakita niya kung paano yung lab procedure, kung paano siya ginagawa in real life. So, in case na nag-imagine lang tayo, di ba? Ayan. So, next would be DNA probing. So, DNA probes are small segments of the DNA that are designed to attract other complementary pieces of DNA in a biological system. So, the small segment of DNA is usually attached to a mounting structure, usually stage yun. Stage or, alam niyo yung sa centrifuge, yung parang tube, ganon. Usually ganon. So, the single-stranded DNA probe attaches to the structure on one end, leaving the other end free to bond to complementary strands of DNA in the solution. So, single-stranded RNA, meron tayong 3' 5' end. So, yung DNA probe nag-attach lang dun sa isang end of one structure, yung isang end ay free. So, first step ay treat natin yung sample with detergent and enzymes para ma-remove yung non-DNA materials. Kunwari sa blood serum, madaming hindi DNA doon na materials like plasma and all. So, kailangan natin silang ma-destroy. So, next would be denaturation. This time, hindi heat ang gagamitin kundi acid solution. So, the solution containing the denatured DNA onto the probe structure, the exposed probe DNA will hybridize with a complementary strand of a single-stranded DNA in the solution. So, i-denature natin with acid solution, magiging single-stranded DNA, tapos ipaprobe natin siya. So, yung unbounded DNA is detected by various methods, such as fluorescence. For example, na-discuss na natin yung fluorescence last time, di ba? So, in each case, it is named fluorescence in situ hybridization or FISH. Ayan, baka makita nyo to pag nag-work kayo sa industry. Yung FISH. So, when using a FISH probe, so if the region of interest is present, the probe will bind to the target DNA sequence. So, naka-code yung target DNA sequence, which can then be visualized using fluorescence microscopy. So, pag nagamit na ng fluorescence, magpo-fluoresce, makikita nyo sa microscope, kunwari yung chromosome, na-cleave natin, nilagyan natin ng probe, tapos yung sample ilagay natin sa microscope, magpo-fluoresce siya pag bombarded na ng light. So, indicating the presence of kunwari, yung target natin na sequence or yung gene or centromere kung saan mo gusto or any region of interest, ayun, makikita natin doon. So, any region of interest na deleted, hindi magbabind doon yung DNA. So, hindi siya ma-visualize. Ayan. So, that is DNA probing. So, next ay DNA microarray. Class yung DNA microarray tsaka yung kanina yung yung PCR, polymerase chain, ay pawang mga applications ng Southern blotting. Mamaya i-discuss ko yung Southern blotting susunod nito. Sana yun yung inuna ko though. So, Sanger sequencing uses different colored fluorescent labels. So, ito yung ginagamit dito, Sanger sequencing. So, DNA microarrays are single microscope slides that arrange tiny amounts of hundreds or thousands of gene sequences on their surface. So, merong normal cell, merong cancer cell, gagawa ng cell culture. Hindi ko alam kung marunong gumawa ng cell culture, pero hindi ko na di-discuss dito. Gagawa ng cell culture, i-isolate yung RNA, gagawa ng complementary DNA. Tapos, paano ginagawa yon? Reverse transcription. Sana naalala nyo pa sa biochem nyo. So, we attach different colored fluorescent nucleotides to samples from the patient's tumor dun sa cancer cell. So, if a gene is very active, there will be a very bright fluorescent area in the gene. So, some genes are not quite as active. So, kung hindi siya ganun ka-active, dimmer or hindi ganun ka linaw, hindi ganun ka intense yung fluorescent area. So, sometimes walang fluorescence, so inactive yung gene. Ganun. So, scientists like us, katulad natin, combine the tumor and normal samples together to determine whether a gene is expressed or not. So, katulad nito, meron tayong healthy cell at saka cancer cell. Kunwari, andun sa cancer cell, naggawa tayo nag-probe tayo ng fluorescent na area na coded yung gene doon. Tapos, after scanning, nag-fluoresce siya. Ibig sabihin, active yung gene na yon. Ay, yung yung area na yon. So, active yung gene doon sa area na yon. Active yung gene doon sa cancer cell na yon or ganun. So, very active siya. Pero doon sa normal cell, wala siya doon. So, makikita natin ngayon, doon natin makita kung ano yung point of natin. Kunwari, sa treatment, anong possible treatment natin doon sa cancer cell para ma-defeat natin siya, para ma-induce natin yung apoptosis o yung cancer cell death. Ayun. Siguro pag nag-med kayo, mas maano nyo. Pero itong method lang muna yung di-discuss natin ngayon. Anyway, so next would be Southern blotting. So, Southern blotting. So, Southern blotting is used to detect a specific DNA sequence. Naturo ko na sa inyo last time yung Western blot. Meron din tinatawag na Northern blot. Ngayon, merong Southern blot. Di ba? May directions. Pero walang Eastern blot. Amazing. A Southern blot test is used to detect specific DNA sequences and samples. So, DNA fragments are separated by electrophoresis. then transferred to a filter membrane where the fragments are detected by DNA probes, katulad ng fluorescence kanina. So, first step, restriction endonucleases cut the DNA strands into fragments. So, yung DNA restriction endonucleases gagawa ng fragments, ilalagay sa agarose gel. So, electrophoresis. Again, yung separation through difference in molecular weight. Kung anong unang mas mababa, siya yung mas mabigat. So, ano yung nasa taas, siya yung lighter. So, electrophoresis on agarose gel separates the fragments by size. So, sheets of nitrocellulose, katulad ng pinakita ko na video last time, membrane, is placed onto the agarose gel containing the DNA fragments. So capillary action moves the DNA to the membrane pababa nung sa nitrocellulose cell. So ionic interaction allows the DNA to bind to the membrane. So DNA ay negatively charged and the membrane is positively charged for them to attract. Ayun, alalahanin yun. Malay nyo lumabas sa boards. So, the membrane is baked in an 80 degrees Celsius, katulad ng last time, for 2 hours to permanently attach the DNA to the membrane. Then, the DNA membrane is exposed to a probe. The membrane, I say, hindi DNA membrane, kasi attached lang siya dun sa membrane. So, the membrane is exposed to the probe. So, the excess probe is washed with the membrane, usually with a buffer, and the pattern of the hybridization is examined. Then, yung pattern, ma-reveal natin yung DNA, yung sequence sa DNA fragments na present dun sa original sample. So, that is Southern blot. So, sana mag-work tong video this time para makita nyo kung paano siya ginagawa. Sana mag-work yung video this time. Okay. Stop presenting. Sana mag-work. Sana. Wait lang daming ads eh. Diba? Daming ginagawa sa biochem, no? Kaya nakakatamad. Anyway, okay. Okay, so yan yung overview ng Southern blotting. So ano ba yung applications ng ito, ng mga inaral natin, yung molecular diagnostics? So genetic diseases can be produced by abnormal number of chromosomes or by abnormality in chromosome structure. Depends. So abnormal number may occur if one or specific pair of chromosomes in an individual is missing. Ayun yung tinatawag nating monosomy. Tama ba ako? Missing. Oo, missing, monosomy. Or if one or a pair, tama ba? One or a pair is present in duplicate. Yun yung trisomy. So, example ng monosomy ay yung Turner syndrome. So, yung female is born with an only X chromosome instead of 2. So, yung trisomy 21 naman, yung Down syndrome. So, trisomy yun. Yung pair is present in a duplicate. Yun yung trisomy, monosomy, individual chromosome is missing. So, the individual is born with 3 copies of chromosome 21 rather than 2. So, yun yung trisomy 21. So, there are many variations of structural abnormalities of chromosomes, including deletion, duplication, inversion, substitution, and translocations. So, in deletions, yung first part na tong image na to, sa upper right, yun, in deletions, part of the chromosome is deleted. Duplications, there are extra portions of chromosomes. In inversion, nagkapalitan sila. Substitution, lumipat siya. Tama ba? Ayun. Sa translocation ay pwedeng reciprocal or non-reciprocal. Tama. In translocation, a portion of one chromosome is transferred to another. So, ayun yung translocation. So, a portion lang. Yung substitution ay substitute. So, ayun. Basically, yung yung term, yun din yung kanyang ang tawag na yung tamang term, variation. Kung ano yung term, yun yung kanyang variation. So, deletion, duplication, oh shit. Ay, hulog yung tablet ko. Sorry, nahulog yung tablet ko. Anyway, in deletion, delete, duplicate, invert, substitute, tapos translocate. So, ayun, yun yun siya basically. So, many times, there are abnormality occurs in the egg, dun pa lang sa egg, or dun sa cell, before conception pa lang, or during conception. Madaming parts ng birth cycle process na pwedeng magkamali. Maraming parts na pwedeng magkamali. So, pwedeng mangyari. Pwedeng mag-delete, pwedeng mag-translocate or what. Tapos mag-perform ng abnormalities like walang legs, walang kamay, ganyan. So, good thing ngayon, nadidetect na natin. At ito nga, yung pinapag-aralan natin ngayon, molecular diagnostics. So, ito yung ginagamit pang detect. Also, another good thing, meron na tayong booming technology to quite correct this. Sana mag-boom. Yun yung tinatawag natin CRISPR. Ayun. Yan yung video na andyan. Siya yung CRISPR. Gusto niyo bang panoorin? May sound kasi to eh. I guess, email ko lang sa yung PDF, panoorin nyo. So, ayan. Yung CRISPR, recently, 2 years ago ata, it won the Nobel Prize Award. So basically, pwede na tayong magkaroon ng way para mag-insert dun sa gene, dun sa chromosome. Pwede na tayong mag-insert ng sequences. Pwede nating i-correct yung sequences through CRISPR. Ayan. Sana mag-boom siya and ma-apply ng sana, para ma-prevent na natin yung mga ganito diseases. Okay. Safari. Stop presenting. That's it. Yun na yung part 2. So, part 3, then exam. Okay. Ito na yung end ng part 2 nung chapter 2 nung sa syllabus. So, after part 3, mag-exam tayo. Gusto mo ba January exam o December na para tapos na? Ano? Hello?
Online po ulit, sir.
Oo. Or ano, kung gusto nyo, face-to-face okay lang naman. Papabigay ko na lang sa mga mga papaproctor ko na lang kung gusto nyo face-to-face exam para wala kayong problema. Kukunin ko lang sa LSP yung exam nyo. Ano? Sa tingin nyo? Yes? Hello? May kausap ba
Online na
ako?
lang po ulit, sir.
Ano?
Online na lang po ulit.
Online na lang ulit. Yung iba, anong nyo? Anong say nyo?
Online na lang po.
Online po.
Online. Okay. Sige. Okay. I guess. Sige. So, December or January? December. Para masaya yung Pasko nyo. Hello? Hello? May kausap ba ako?
Nagde-decide.
Okay. Anyway, chat nyo na lang kung ano mapagtripan nyo. Yung January, ano? December 24 ng 12 midnight. Okay ba yun? Joke lang. Defense ko kasi sa 14. So, probably after that, after December 14 sana. Ayan. Okay ba yun? Like after December 14. Week after
Pag-usapan po muna namin, sir.
Oh, sige. Basta hindi dun sa week ng December 14 kasi defense ko yun. So,
Okay po.
I guess baka
Good luck po, sir.
salamat. Baka butaw-butaw pa akong doon. So, salamat. Okay. Salamat sa pagpunta.